Great, thanks. It's nice to have an audience where you know everyone by first name. So I want to start by thanking Paul for putting all this effort into developing the whole NEUROGUT concept, but also managing the process and the program, and being a leader for all of the fellows who were taken on to this project. I think we all appreciate the efforts that he's made and are very grateful for that. What I was going to do is tell you a little bit about the science underlying the project that we did in Sheffield. When Paul asked me to do this, I wasn't sure whether he wanted me to review all of the science of all of the projects, and didn't feel comfortable with that, and also felt it would be so superficial as to not be terribly effective. So I'm going to go into some of the details around the project that we did in Sheffield, and provide you with some of the data that Frederica has generated over the last 3 years. She has a poster on tomorrow, and so I would encourage you to go and talk to her at a poster to get more insight and more detail of what we did there. So really what Paul asked me to do was to give a perspective from a PI. I don't really need to go into the whole project. I think the point that I was going to make here is that this was 4 years in development. And so many of the ideas for scientific projects that were discussed in the original period in 2009, by the time the work was funded, much of that work had already been done. And so one of the advantages of these training networks is that you're not tied to specific work packages in the same way that you would be as part of a framework program collaborative award. And so that provides you with a great deal of flexibility. And indeed, when we initially started the work with Frederica, we were going to look at luminal signaling from the perspective of nutrients and endocrine cell function, but very quickly took advantage of some opportunities that we had in Sheffield around bacteria, and that led us into a slightly different area from what was originally outlined in the project, and I think has developed into a very fruitful project. I don't need to go through this. This is what Paul's just presented, really just to highlight the three areas that Paul has described, and to say that in Sheffield we were training package 7, and we were sitting there with interactions with Michael Schiemann in Munich, Guy Buchsteins in Leuven, and others in Bologna. So we were sitting in this group of projects that was interested in visceral sensory signaling and the role of inflammation in hypersensitivity. What I would just comment on, given the events in the UK in the last 12 months or so, is that opportunities for Britain to take part in these kind of training networks may be jeopardized by Brexit. And here's what Europe might look like in terms of funding once we've gone through the formal process of pulling out of the European Union funding block. All the fellows are there on the front row. Here they are at one of the annual meetings. This one held in London at the Wingate Centre. And you can see at the front all of the research fellows. At the back you'll see some of the PIs. I don't know is it the pointer? So André Smout there, Guy Buchsteins, Magnus, myself, and also some of the external advisors, so Christine finally, and Jack Wood. And one thing that I will say about this group is that it was a very collegiate group. The fellows all seemed to get on with each other. They were happy to help each other to develop ideas and develop concepts. Through the frequent training program workshops that they had opportunities to work together, and I think formed a very cohesive group. The other thing I would say is that the PIs were also very collegiate, and this perhaps reflects the fact that many of these PIs had collaborated previously. So this was an amalgamation of groups who had proven to work effectively together for quite a number of years. And I think that collegiate environment was really important in the way that NEUROGUT developed. And I can illustrate that by comparing to another training network that I was involved in some years back, based on urology research. And in that network, there was a lot of negative interactions amongst PI. It was very competitive. And quite often, that was taken out on the research fellows. And so, as a consequence of that, it was a very different atmosphere to what we had within NEUROGUT. And I think the consequence of that was that at the end of this urology training network, not one of the fellows stayed on in urology research. So I think it defeated the object of the project. And I suspect, although I've not spoken to all of you individually about that, but I suspect that many of you will be staying on in the GI research field and have the potential to be some of the rising stars of the future and make an impact on the field. So I think from a PI's point of view, to be able to witness that growth and development of you fellows, I think has been very gratifying, and I think is a measure of the success of NEUROGUT. I think the success will also be measured in terms of research outputs, although they're not a critical part of the assessment. But I know that there's publications already in place and others in planning that will carry the NEUROGUT logo and demonstrate not just the effectiveness of the training, but also the effectiveness of the scientific projects that have underpinned that. As a training network, the funding is very different from the funding for collaborative awards, in that individual projects aren't costed. There's a set amount of money that follows each of the research fellows. Most of that goes towards personal stipend and training needs, but there's an allocation for the PI's laboratory. But for expensive projects, that allocation of funds is not always sufficient to fund the research. So it's not a lucrative network in terms of personal research funding that drives people's willingness to take part in these training networks. But what it does provide is an opportunity to have very bright, gifted fellows work in your lab and to develop the work. And I think that's something that we've all the PIs will have appreciated and gained from that. So that's my sort of personal perspectives on this. I thought I'd say a little bit about the science that we did, and the science was based around hypersensitivity and the fact that the relationship between a stimulus and the response in terms of suffering, pain, isn't fixed. It can be upregulated in response to infection and injury, and this leads to what is called hypersensitivity. And as part of that hypersensitivity, stimuli that would normally be unperceived now become perceived, and this is referred to as allodynia, and painful stimuli give rise to more intense painful experiences, and that's hyperalgesia. And these are the hallmarks of visceral hypersensitivity, allodynia and hyperalgesia. The concept that has developed, particularly around post-infectious IBS, is that some kind of triggering event, some inflammatory episode, gives rise to changes within the intestinal wall that lead to immune activation and long-term changes in sensory signaling. And we've, over a number of years, been interested in trying to understand what is the molecular basis of that upregulation and hypersensitivity. And the concept that has been developed and was tested as part of NEUROGUT was that sensitization involves both peripheral and central mechanisms. We were more interested in the peripheral. Kazim and Paul were developing ideas around central sensitization. But in the periphery, what is thought to happen is that the chemical environment within the gut wall changes as a consequence of an immune or injurious response. The chemical environment contains many mediators that can influence directly the sensitivity of nerves, or can interact with immune cells to develop a cascade of responses that gives rise to an altered chemical environment. And this is often referred to as an inflammatory soup or a sensitizing soup. And this is something that is now recognized in patients with IBS, and I'll show you some evidence for that. But there are many different mediators that are involved in that sensitizing soup that interact with ion channels and receptors on the nerve terminal to drive sensory barrage into the nervous system, where you have activation of pain pathways, sensitization of those pathways, or recruitment of what are referred to as silent nociceptors. And these are nerve endings that normally don't give rise to pain, but they're normally silent, they don't give rise to any response, but in response to inflammation and injury, they become awakened and contribute to that sensory signaling. On top of that, you have changes in the central nervous system, in the spinal cord, where you have an upregulation of synaptic strength, so the information is transferred into the spinal cord more readily, and you have changes in the brain that interpret those signals in a different way in patients with these chronic IBS symptoms. Our focus was on the periphery, and in the periphery we can identify different types of sensory nerve endings that are really characterized by where the endings terminate in the gut wall. So we can talk about mucosal afferents, we can talk about muscular afferents, muscular mucosal that have bifurcating endings in different regions. We've got endings around the enteric nervous system that are referred to as intraganglionic laminar endings, others within the serosa, particularly associated with blood vessels. And these endings in different layers of the gut wall respond to different mediators, different types of stimulus, to provide a rich repertoire of sensory signaling from the gastrointestinal tract. We experimentally tap into those afferent nerves by attaching them to recording electrodes as they emanate from the bowel wall in what are called mesenteric nerve bundles. And we can attach those to electrodes, and that allows us to record the action potentials generated in these nerve endings en route towards the central nervous system. We can do this in vivo or in vitro. And these spikes you can see here are the action potentials in different afferent nerves. And each nerve, the action potentials from each of these afferents, has a very characteristic waveform. And what that means is that we can extract, using waveform detecting software, individual single units from within this multi-unit preparation. And that allows us then, through a series of different stimuli, to compare the properties of these different nerve endings terminating in the gut wall. Now, one of the features of hypersensitivity is that the relationship between the stimulus and the response is increased. And this is a recording from one of these nerve bundles supplying the mouse jejunum. And what you can see is that as you increase the pressure within the loop of intestine, the action potential discharge increases. And it increases in a biphasic way. You get a very rapid increase of low changes in pressure, which then develops into a gradual change as the pressure increases. And when you get up to these levels of 30 and above, what you're dealing with is pressures that in individuals, in you or I, would give rise to pain. So these are the nociceptive signals. These are low threshold afferent activity that would normally be activated as part of physiological responses to, for example, ingestion of a meal. So this is reflecting the whole sensory repertoire across that mechanical threshold range. If you take one of these animals that has become sensitized as a consequence of an inflammatory response, in this experiment we've used a nematode worm called Trichinella spiralis, which we use deliberately because it produces a very transient inflammatory response, and then the worms are expelled. So by the time we make our recordings 4 weeks after the initial infection, the worm is long gone, but the consequences of the inflammation have been maintained. And so this is a model of post-infectious hypersensitivity. And you can see that hypersensitivity reflects both low and high threshold components of that afferent signaling pathway. Over the years, we've looked at the mechanisms behind that using a variety of pharmacological tools, but also transgenic animals. And this is an example of one set of experiments that were performed using the P2X7 knockout mouse. And P2X7 is an important receptor for ATP generated during inflammation and injury. And it's a unique receptor in that it forms at high concentrations of ATP a pore in the membrane, allowing large molecules into and out to cells. It's a feature of immune cells. So the P2X7 receptors are expressed on macrophages, on lymphocytes, and on mast cells, not necessarily on the afferent nerves themselves. So what we see here in the knockout mouse is that the response to mechanical stimulation is unchanged, but in the post-infectious state, we no longer see the hypersensitivity. So what this is saying then is that you can block the inflammation, then you can block the generation of hypersensitivity, demonstrating that link between an inflammatory event and the onset of hypersensitivity. And in these experiments, you can look at various components of that inflammatory response. Here is immunohistochemistry for IL-1 beta, and there's an upregulation of IL-1 beta in the wild type, but not in the P2X7 knockout. And we can also look at cytokine profiles. And these are blunted in the P2X7 knockout. So it's demonstrating that link between inflammation and hypersensitivity and pain. And that's where we started to think about whether those chemical environments, that inflammatory state in the gut, could be underlying symptoms in patients with IBS. And Giovanni came up with the idea of taking biopsies from these patients with IBS, extracting the soluble mediators from those biopsies, taking the supernatants from the incubating medium, and applying that supernatant to tissues and cells. And remarkably, when you apply supernatant from biopsies taken from IBS patients, you can drive that afferent sensitivity, whereas supernatant from healthy controls had no effect. So supernatants from these patient samples predisposes to sensitization. And I think this was the first demonstration then that there is a chemical change within the gut wall. And that has, I think, resulted in a paradigm shift with regards to thinking around IBS, that rather than being a functional disorder where you cannot identify changes within the tissue and periphery, here we have a biomarker of hypersensitivity in patients, demonstrating that there is a change within the gut that predisposes towards heightened sensory signaling. And since then, many groups have taken samples from different patients with IBS, with IBD, supernatant from feces, from biopsies, and looked at the chemicals that can contribute to that. And I think it was that first paper from Giovanni that has driven that viewpoint, and it's been replicated many times, implicating different mediators in that hypersensitivity. So in our study with Giovanni, we focused on histamine, and histamine H1 receptors was shown to have a big effect. In other studies, people have found proteases contribute enormously to that hypersensitivity. Others found 5-HT. Different receptors, different mediators, all contributed to that hypersensitive state. Michael Schiemann and Guy Buchsteins and I also looked at supernatants from IBS patients who had had Barostat investigations in order to look at the relationship between their sensitivity, so quantifying their hypersensitive state, and the ability of supernatants from these samples to drive firing. And what you find is that patients that are hypersensitive are able to produce a much greater neural response than IBS patients that are normosensitive, that have a normal threshold for Barostat distension. Again, confirming this relationship between the chemical state within the gut wall and the ability to activate afferents. This is in dorsal root ganglion neurons, so in primary sensory neurons, but Michael also looked in enteric neurons using his voltage-sensitive dyes, and again you see the relationship between hypersensitive samples producing a much greater response than normosensitive samples. Around the time that we started on NEUROGUT, we were beginning to become interested in the microbiota. And there had been a number of studies, particularly from Leonor Bueno's group, taking fecal samples from hypersensitive animals and patients and showing that they could influence afferent firing. And this was also a major theme within NEUROGUT. So many groups were interested in pre- and probiotics and the influence that this can have on sensory signaling. But in Sheffield, we took a different view. Rather than thinking about the beneficial effects of commensal bacteria, what we wanted to know was whether pathogenic bacteria might influence afferent firing. And our thinking along those lines was triggered by this paper in Nature published from the group in Harvard. And what they showed was that if you inject Staphylococcus aureus into the skin, then you developed a peripheral hypersensitivity. You developed a pain state. But what was different about this from all the other studies that have looked at hypersensitivity in cutaneous pain was that they found a direct correlation between the bacteria and the generation of the pain state. The thinking before here was that the bacteria trigger an inflammatory response, and the inflammatory mediators then gave rise to the hypersensitivity and pain. But what they showed here was that, first of all, that Staphylococcus produced this time course of a pain response. It was absent in transgenic animals that had their NaV1.8 containing sensory neurons deleted. But this is the important slide that looks at the relationship between pain in red and the various other parameters that they measured, including tissue swelling, immune cell recruitment, and the important thing is this correlation between bacterial load and pain. So this suggested that it was the bacterial load rather than the inflammatory response to that was driving the hypersensitivity. The bacterial load and the immune response came after the hypersensitive state had peaked. So we wondered whether we could investigate those things in visceral afferents, with the rationale that in some patients you get gut infections with Staphylococcus aureus, and this can lead to severe inflammatory damage and disease, including sepsis and death. One of the other reasons for thinking about Staphylococcus aureus is that there's a huge library of different strains of Staphylococcus that have mutations that govern their virulence and toxin generation. And we were able to utilize, through a collaboration with Simon Foster in Sheffield and then other colleagues that he put us in contact with, to access these different strains of Staphylococcus aureus. And we looked at one here called J2. This is our, if you like, our wild type strain. This is one of the strains that's involved in MRSA. But we used another strain here, Newman SH100. And these differed from each other in terms of the way these cells respond to external factors, leading to the release of toxins. And so these different strains are controlled differently by various quorum sensing mechanisms. And one of the master regulators of quorum sensing is AGRA. There are other factors that also respond to what is called autoinducers, and these detect other bacteria. They detect the density of bacteria within the particular environment, and they, through this activation of AGRA, release a whole range of different toxins and virulence factors. And some of the ones that we were interested in were alpha-hemolysin. This is a toxin that punches holes in cells, leading to influx of calcium and ultimately cell death. But Staphylococcus also releases various proteases and various other molecules that can interact with cells involved in immune activation, but also potentially interact with neurons, such as phenosoluble modulins and formal peptides. And these different strains then produce different balances of these mediators and toxins. So the first thing we wondered is, if we apply supernatant from our growth plates of Staphylococcus aureus, does that have any effect on the nerves? And so Frederica developed this technique where she took a clone of Staphylococcus aureus, incubated it overnight, then did a 1 in 100 dilution and grew the bugs up for a period of time after that, sampling the supernatant and examining its effect on visceral afferent sensitivity. And the idea behind this, the principle, was that as these cultures grow, they shift through this experimental growth phase, exponential growth phase, to a stationary phase. And during that phase, they shift from expressing various quorum sensing molecules on their cell membrane to producing secreted factors and mediators. And so we wanted to know what the relationship was between this growth the soluble factors and their effect on nerves. And here's one example of that. Here's the vehicle added to the nerve, produces a small increase in activation. But when you apply the supernatant from Staphylococcus aureus, here you see a greater increase in discharge. These are the responses to that mechanical distension, so you get a mechanical response. And as you can see, initially there's an increase in baseline firing, so these nerves become activated by the supernatant. But then the mechanosensitivity starts to become blunted, and then eventually the baseline firing falls and mechanosensitivity is completely lost. And as you look at the envelope of activity in this nerve recording, you can see that the nerve has gone effectively very quiet. The Staphylococcus has almost effectively killed the nerve. But we wanted to know what was happening here during this excitatory phase, and then what was happening during inhibition, and what were the mediators that might contribute to that. Here's the biphasic nature of that response, looking at the J2 supernatant. Here's the inhibitory effect on mechanosensitive firing. Just a couple of observations that Frederica made. One is that the inhibitory effect here is dependent upon that growth phase. So only when it reaches the stationary phase do you get this major inhibitory effect. So it depends upon the growth stage of the bugs, and that's true both for baseline firing and mechanosensitivity. And it was also concentration dependent. So 5, 10, 20% supernatant here has a graded effect on both the excitatory and the inhibitory effect. The first experiment to try and identify mediators Frederica did was just to boil the supernatants to see whether these were heat-sensitive mediators, and indeed they were. So both the baseline effect and the effect on mechanosensitivity was influenced by pre-boiling the supernatants. But this, I think, is the most interesting aspect of the work. These are these different strains of Staphylococcus aureus. The J2, this is a wild type. Human has a defect in SAR signaling, which means that there's a reduced amount of toxins produced. SH100 has even less toxins. AGRA produces no toxins. And then there are other mutants that are defective in specific mediators, such as the alpha-hemolysin mutant here. And what you can see here, oops, get used to this. This is the effect on firing at 30 minutes. This is the excitatory phase. Here at 90 minutes, this is the inhibitory phase of the response. And you can see that J2 and human produce an increase. It's much less in the SH100 and it's lost in the AGRA and hemolysin mutants. In terms of the inhibitory phase, again, it's strain dependent. The human and the J2 strain are very different in terms of that inhibitory phase. And then there are marked differences as well in the AGR and hemolysin. So we can start to dissect out the effects of different mediators from these mutants on the excitatory and inhibitory component of the afferent response to the supernatants. Frederica looked at other things as well, and this is a cytotoxicity assay using prapidium iodine fluorescence to indicate pore formation, calcium entry, and subsequently cell death. And you can see that J2 and human have very similar effects on the prapidium iodide fluorescence in DRG sensory ganglion cells. The AGRA mutant, this is the quorum sensing mutant that doesn't produce toxins, has no effect. So clearly this is a demonstration of how you can use microbiology in order to identify different mediators. Frederica also looked at the expression of different receptors on DRG and within the tissue itself. ADAM10 and sphingomyelin synthase are components of the pore formation pathway that occurs in response to alpha hemolysin. So they're important for alpha hemolysin signaling. FPR1, 2, and 3, these are receptors to formal peptides. These are the receptors, GPCRs, that respond to these specific mediators released from these different bacteria. And you can see that there is expression. It's somewhat low for the formal peptides, higher for ADAM10 and SGMLS, and there are some differences depending on where you look relative to the sensory input to the spinal cord. So this is cervical, thoracic, and lumbosacral regions, and nodose ganglia. And you can see that some of these are higher in nodose and lumbosacral. So there's still work to be done to explore the expression of these different receptors in these different tissues. But just to wrap up, what I think we've shown through the NEUROGUT project and through our funding for Frederica's PhD is that this idea that neuromodulation occurs as a consequence of that inflammatory response, the release of inflammatory mediators, needs to be revised in order to consider direct effects of products from bacteria. And so the bacteria themselves can contribute to that altered environment within the gut that could lead to altered signaling. And the important point to stress is that some of those mediators can increase excitability, some of them can decrease. And so a balance of those may be important in setting the neural tone. Frederica's looked at other aspects. She's looked at effects on motility and using Ussing chambers, the effects on ion transport across the mucosa. And there's no consistent pattern between what's happening on the afferents and what happens to motility and secretion. Again, suggesting that different mediators from these different boats can have differential effects on the gut wall. The pore-forming toxins appear to be important for inhibiting peristaltic activity, whereas other mediators seem to have their effect on secretion, some of them acting directly on the enterocytes themselves rather than through the neural mechanisms. And then finally, to acknowledge NEUROGUT funding, obviously, but also the labs involved. So our group in Sheffield, together with Simon Foster, who's the microbiologist with special interest in Staphylococcus aureus. There's Michael's group in Munich, where Frederica went and spent a few weeks as part of the collaborative exchange element of NEUROGUT. And then also Stuart Briley's group in Adelaide. Frederica has just come back from a few months in Adelaide, having Adelaide as a recognised NEUROGUT associate partner, allowed Frederica to go there to do some elements of the work. And that, again, was another important aspect of the NEUROGUT project, to allow people to go and exchange between labs and broaden their experience of research. So thank you.